Sorry about being late. I had to verify my existence even though I had logged in for the lab earlier, so that sort of surprised me. Before we begin, are there any questions? All right, share my screen here. Should be seeing my screen now. Today is October 30th, and we should be starting chapter 6. I don't think we're there yet. Any questions about any of that? Let me look up where we are in chapter 5. Let me remind you that once I finish a lesson, I will post the slides. If the slides are not up, please remind me to get them posted. All right, we're on slide 58. I talked about starting fermentation, shown on the right. Fermentation is another way that cells can obtain energy for making ATP. Let me blow this slide up. Fermentation always begins with glycolysis, making glucose into pyruvic acid. That's what makes the ATP in fermentation, by the way. It does make NADH, but the NADH is used in the fermentation step, either one or two chemical reactions after glycolysis, to make the fermentation end product, like lactic acid fermentation, making lactic acid. Any question about that? The difference between fermentation is it only makes 2 ATP molecules per molecule of glucose, much less than the 36 to 38 molecules of ATP that aerobic respiration can generate. So fermentation releases energy from oxidation of organic molecules via glycolysis, meaning largely from glucose. In glycolysis, you get out of glucose two pyruvic acid molecules, also called two pyruvates, two net ATP, an NADH molecule. Do realize that other molecules can be used besides glucose, but usually these molecules are converted into glucose and then fermented in the usual way. All right, fermentation does not require oxygen, so it can happen in the presence of oxygen or in the absence of oxygen. It does not use the Krebs cycle or the electron transport chain. It does use an organic molecule as the final electron acceptor, meaning the electrons flow to an organic molecule which is usually pyruvic acid. That's where the electrons go. And then the electrons, that molecule is converted into the fermentation end product, which for lactic acid fermentation would be lactic acid. And that will be just one chemical reaction after making pyruvic acid. There are two fermentation reactions I want you to know. There's lactic acid fermentation. This is something that your muscle cells can engage in, where glycolysis of glucose produces 2 ATP, and then there's one fermentation step further, converting the pyruvic acid into lactic acid. This is a fairly common chemical reaction in living cells because a food spoilage organism could make lactic acid. Sauerkraut uses lactic acid fermentation. Yogurt uses lactic acid fermentation. Most cheeses use lactic acid fermentation. like cheddar cheese, for example. Yogurt, I already mentioned that one. Sauerkraut uses, oh, I got that there. Never mind. Chocolate uses lactic acid fermentation. Another fermentation reaction you should know about is alcoholic fermentation, also called ethanolic fermentation, where pyruvic acid is changed into ethanol. Pyruvic acid is a three-carbon molecule. Ethanol is a two-carbon molecule. So CO2 comes out of this reaction. And like I said, this one takes two steps to make um, the alcohol. The CO2 coming off is actually what gives beer its fizz. Wine does make CO2, but it is made in an open barrel or tub, and then the CO2 just vents to the atmosphere. If you were to make the wine in a bottle and cap the bottle, the CO2 would be collected. Let me go back. In the bottle of wine, and that we call champagne. Any question about any of that? There are more than one or two fermentation reactions. Like I said, I'm only asking you to know lactic acid fermentation and alcoholic fermentation. Alcoholic fermentation almost always happens by a yeast, Saccharomyces cerevisiae, I think. And that's what makes leavened bread, by the way. When you put in the yeast to make leavened bread, it also is what we use to make beer and wine. There is another fermentation reaction which actually makes Swiss cheese, propionic acid fermentation, and it does give off CO2, and that is why Swiss cheese has holes in it. Any question about that? There are other fermentation reactions. I think this one is the one they use to make vinegar. This is one that is used in industrial purposes to make butanol or isopropyl alcohol. Any question about any of that? So there is more than one fermentation reaction. I'm just asking you to use know the lactic acid fermentation because it's very common. Most of our fermented food uses lactic acid fermentation. By the way, sourdough bread uses both yeast and lactic acid fermentation to make the sourdough bread. So you do get the CO2 from the yeast to leaven the bread, but it's sour, and so it's not really the yeast making it sour. It's partly the yeast, but it's partly the bacteria making the sourdough bread sour. And this is some industrial uses of different type of fermentation reactions. Beer, wine, and fuel uses ethanolic fermentation. This is also the one we use for bread, by the way. Vinegar using acetic acid fermentation. Um, Cheese, yogurt, rye bread, sauerkraut, summer sausage, chocolate. Most fermentation, and I should say cheddar cheese because there's an exception on one cheese, but almost all cheeses use lactic acid fermentation. The exception is Swiss cheese, which uses propionic acid fermentation. And these others I'm not going to even talk about. Any question about fermentation? All right, so this is a good summary table for respiration, both aerobic respiration and anaerobic respiration, comparing them to fermentation. Aerobic respiration must happen around oxygen in the air, so that's why it's aerobic. Anaerobic respiration does not require oxygen and generally happens in the absence of oxygen. The final electron acceptor for aerobic respiration is, of course, oxygen. The final electron acceptor for anaerobic respiration is a molecule other than oxygen. And then fermentation can happen either under aerobic or anaerobic respirations, and it uses an organic molecule as the final electron acceptor. The generation of ATP, you can skip this one here. The generation of ATP, aerobic respiration is the most efficient. Prokaryotes generating 38 ATP molecules per molecule of glucose. Eukaryotes, most cells generating 36, but a few cells generating 38. Anaerobic respiration will be variable depending on what the final electron acceptor is or what form of anaerobic respiration it is. It'll always be fewer than 38, but more than 2. The most efficient anaerobic respiration can only generate 36 molecules of ATP per molecule of glucose. Fermentation always generates 2 molecules of ATP per molecule of glucose. Let me mention with fermentation, when you work your muscles and you have muscle burns, that is from the lactic acid building up in your muscle cells. Any question about any of that? All right. So I've talked about glucose being used in catabolism to make ATP, like with the aerobic respiration, but other carbohydrates can be used. Most of these carbohydrates will simply be broken down and then converted into glucose. And then glucose will be sent down aerobic respiration in the normal way to make ATP. Other molecules can be used, like lipids and proteins, but there is a problem. This is true for large carbohydrates too, and that is these large molecules cannot cross the cell membrane and then enter the cell. So how cells get around this problem is they secrete extracellular lipases and proteases to digest the large molecules outside the cell, break the large molecules down into the monomers, and then bring the monomers into the cell. Like proteins, this is the way our human body digests proteins too. We secrete extracellular proteases, digest the protein into amino acid, and then bring the amino acids across the cell membrane using transport proteins. With lipids, you do the same thing, and so do microbes. They secrete lipases extracellular, break them down into the monomers, meaning the fatty acid, lipids, and the glycerol, and then bring the monomers into the cell. Any question about any of that? All right, let's talk about the interrelationships of carbohydrate, lipid, and protein catabolism. I've talked about it, that they are independent. That's not entirely the truth. It is true that proteins will be digested into amino acid, and then depending on the amino acid, some of them will enter late in glycolysis, some of them will be converted into acetyl coenzyme A and then enter the Krebs cycle. Others will be converted into a late intermediate of the Krebs cycle and then enter the Krebs cycle. Lipids will be broken down into glycerol and fatty acid. The glycerol will enter glycolysis midway through and then be digested. Fatty acids will be broken down into 2 carbon chunks and then converted into acetyl coenzyme A, and enter the Krebs cycle there. Anyways, this is how proteins, carbohydrates, and lipids are digested. Carbohydrates, I already mentioned, are essentially converted into glucose. All right, most cellular ATP is used for the synthesis of new molecular components, meaning anabolism, but ATP can be used for any of the cell's needs. And this ATP in our cells comes from the metabolic oxidation of glucose or some other molecule which will be converted into glucose. And here I'm talking about anabolic and catabolic pathways are not independent. They do have common intermediates joining the two pathways. We call them amphibolic pathways, a metabolic pathway that can function in both directions, meaning you can, let's see, uh, you can digest carbohydrates and make glucose and then send glucose through aerobic respiration the normal way, but you can also take glucose and then make a carbohydrate. And this is true for lipids. You can burn lipids, but you can also take glucose and build up lipids. I think everybody actually knows this internally. What happens if you eat too much sugar, such as too much glucose? More than your body can burn to make ATP. What'll happen? Anyone, if you eat too much glucose? Are you simply not going to gain weight? Actually, a few of you, if you have a metabolic capacity like I do, I don't gain weight or didn't gain weight until recently. But I'm not sure what happened. But no matter how much I ate, I never gained weight. But most of you, if you eat too much glucose, what's going to happen? Come on, somebody's got,
Gain weight?
yeah, you gain weight. And is that going to be put down as carbohydrate? You're,
No, fat.
it's going to be converted into lipids or fat. And so that's telling you that um, glucose or any other carbohydrate, if you eat too much of it, it will be converted into lipids and then stored in the body as fat. And so you already know that about amphibolic pathways, that carbohydrates can be converted by your body into lipids or fat. That's true for proteins too. This reaction is showing you how this happens. Let's say we have an amino acid from digested protein and it enters the Krebs cycle here. Well, surprisingly, your body may not digest it in the normal way, that it may convert this amino acid into glucose. How? By going backwards, 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 backwards. That's a pathway in two directions, going back to acetyl-coenzyme A, using acetyl-coenzyme A to make pyruvic acid, going this way. And you see this here? This is a two-way pathway because it got the arrow there. So glyceride 3-phosphate can be converted into fructose 1,6-diphosphate and then go and make glucose. For most of us couch potatoes, if we were to eat too much protein and too much lipid as well as too much carbohydrate, uh, the protein and the lipids will be converted into glucose and then sent down aerobic respiration in the normal way. Any question about any of that? And how that happens is the amino acid or the lipids here will be sent here and then go back this way to make glucose. Or the amino acid here will go back a very long way to make glucose. Now, that's not the case for a marathon runner. A marathon runner can burn lipids directly in aerobic respiration, but a marathon runner will not burn lipids until 45 minutes into their run. For the first 45 minutes, they will either not be digesting lipids or they will be converting lipids into glucose and then sending glucose down aerobic respiration in the normal way. It's not until 45 minutes or later in the marathon run that a marathon runner will actually burn lipids in aerobic respiration. Any question about any of that? And I would assume it's something similar for proteins. I don't know the numbers, but like I said, initially, the body will not burn proteins. In aerobic respiration, it will convert the protein to amino acids, convert the amino acids into glucose, and then send glucose down the normal way in aerobic respiration. And that's the way us couch potatoes do it. I'm saying I'm a couch potato because I don't go out and do heavy exercises anymore, and I certainly don't run in a marathon run. Any question about any of that? All right, let's briefly talk about photosynthesis. Photosynthesis is the conversion of light energy to chemical energy. The chemical energy may be used to convert atmospheric CO2 to glucose. So oftentimes, photosynthesis is used to make glucose. Not all the time, but a lot of the time. And this conversion of atmospheric CO2 to glucose is called carbon fixation. If you don't know, all living life on the planet requires glucose, which we get mainly from photosynthesis. The only exception would be like a deep sea vent where there is no sunlight reaching it, and that deep sea vent, the ecosystem is running by chemosynthesis, not from photosynthesis. But all other places on the planet, photosynthesis is the primary producer of food. Photosynthesis is performed by cyanobacteria, algae, and obviously green plants. And a mechanism for making organic compounds such as glucose, which animals and other microbes feed on. Photosynthesis only happens in photosynthetic organisms. These organisms contain a light-trapping pigment. One of them you know is chlorophyll. What happens is light releases electrons from the chlorophyll, and then that light energy is converted into chemical energy via an electron transport chain. ATP and NADPH, that's similar to NADH, are always generated, but oftentimes ATP and NADPH will be used up to make glucose from CO2 and water. And that's the carbon fixation reaction. But there are two types of photosynthesis reactions, a cyclic photosynthesis reaction and a non-cyclic photosynthesis reaction. I'm sure in your preliminary biology course, they taught you the non-cyclic photosynthesis. They just never probably told you it was non-cyclic photosynthesis. The summary equation for non-cyclic photosynthesis making oxygen is 6 carbon dioxide molecules get together with 12 water molecules in the presence of light to make glucose 6C6H12O6, 6 oxygen molecules, 6 O2s, and then 6 water molecules. Sometimes you'll see this equation minus the 6 water here and then minus 6 water there. But I prefer this full equation because if you look at where all 12 of these oxygen atoms come from, they all come from water. None of these oxygen atoms come from CO2. All of the CO2 will be converted into glucose. There's another equation happening in anaerobic phototrophs. They do not make oxygen, they actually make sulfur. And you don't need to know this summary equation, but there are two non-cyclic photosynthesis reactions. And then there's a cyclic photosynthesis reaction. In cyclic photosynthesis, the only product made is ATP. So you simply, in cyclic photosynthesis, combine ADP and phosphate to make ATP. All photosynthetic cells can engage in cyclic photosynthesis and non-cyclic photosynthesis. If the plant is wanting glucose, It'll engage in non-cyclic photosynthesis to make the glucose. But if the plant or photosynthetic cell is wanting ATP, it will engage in cyclic photosynthesis. Any question about any of that? All right, let's talk about cyclic photosynthesis, also called cyclic photophosphorylation. Light will hit the uh, electron trapping pigment, or in this case, chlorophyll, and that will excite two electrons from the chlorophyll to leave the chlorophyll. Those electrons will then enter in an electron transport chain, first moving to this protein in the membrane, then moving to that protein in the membrane, then moving to that protein in the membrane, moving back to this protein in the membrane, and then that electron will come back to the chlorophyll. So the electron is traveling in a circle back to the place it started, and that's why it's called cyclic photophosphorylation. Any question about any of that? Well, when the electron moves through this protein in the membrane, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. This will create a twofold gradient. There will be more hydrogen on this side of the membrane than on that side of the membrane. There'll be more positive charge on this side of the membrane than that side of the membrane. These H plus ions, by the second law of diffusion, will want to move over to this side of the membrane. They can only do it by coming through this transport or channel protein. This channel protein is ATP synthase, and when the hydrogen ions move through the protein, they give the protein the energy it needs to phosphorylate and make ATP. Phosphorylate is adding phosphate to ADP, or adding phosphate to some molecule, I should say, making ATP. This should be a little bit familiar to you because it's very similar to the electron transport chain in aerobic respiration, creating a twofold hydrogen gradient. And then the hydrogen gradient, you're using the potential energy of all these hydrogen and positive charges over here wanting to come over here to make ATP. So it's very similar. Let me end the show here to very similar to this reaction in the electron transport chain. The difference is that there's fewer proteins in the chain in cyclic photosynthesis than there is in aerobic respiration. But you do cause the hydrogen ions to form and all that. The cyclic photosynthesis occurs in the chloroplast. Let me blow this up. So it's not the mitochondria here, but it's happening in the inner chloroplast membrane of a eukaryote cell. So this is a eukaryote cell, and this membrane here containing the electron transport chain is the inner chloroplast membrane. And this is the inside of the chloroplast. This is the space between the inner chloroplast membrane and the outer chloroplast membrane. So the hydrogen ions are pumped there. In a prokaryote cell, this is the cell membrane, this is the cytoplasm, and this will be most likely the cell wall if the prokaryote has a cell wall, it's pumping these hydrogen ions into the cell wall. And that's very similar to what's going on in aerobic respiration. Any question about any of that? All right. So in summary, you can have a energy a source varying, meaning the electron source can vary. It can happen, the electrons can come from glucose in carbohydrate fermentation or carbohydrate metabolism and aerobic respiration. It can also come from the sun, the energy of the sun striking the pigments, chlorophyll, for example, causing those electrons to leave the chlorophyll. And then there will be an electron carrier molecule carrying the electrons to the electron transport chain. And then the electron will reach a final electron acceptor. In aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, the final electron acceptor is a molecule other than oxygen. For fermentation, the final electron acceptor is an organic compound of fermentation. And it doesn't have this here. I'm not sure why not. The final electron acceptor in cyclic photosynthesis is chlorophyll, the molecule where the electron started from. Any question about any of this? The point is the electron donor or energy source can vary, and the final electron acceptor can also vary. Any questions about chapter 5? All right, let's move on to chapter 6. All right, in this slide, I'm showing you chapter 6, microbial growth, showing you the major goals and a rough outline. Know the terms of this chapter? What's a psychrophile, a psychotroph, a mesophile, a thermophile, a hypothermophile? What's an acetophile, a halophile? What is an aerotolerant anaerobe, a facultative anaerobe? What is binary fission? What are reproductive spores? And what are endospores? Any question about the terms or the outline so far? All right, 2, understand the various requirements of cells for growth. 3, understand the phases of growth. And then 4, compare the different methods we have of measuring microbial growth. Any question about any of this? When we talk about microbial growth, we are referring to an increase in the number of cells, not in the increase in the cell size. That was different when I used to be teaching in middle school, when we talked about the middle school students growing. Hopefully,
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
all right. As I was stating, when I was talking about the middle school students, when we talked about them growing, hopefully we're not talking about them increasing in the number of middle school students, just the size of the middle school students. Any question about any of that? All right. The requirements for microbial growth are both physical and chemical. Physical requirements including the microbe has to be at the correct temperature, the correct pH, the correct osmotic pressure, meaning the correct salinity. And those are some of the physical requirements cells have for growth. Chemical requirements is the cell has to have a source of carbon, a source of nitrogen, a source of sulfur, a source of phosphorus, a source for trace elements, and a source for oxygen if oxygen is required, and organic growth factors if they are required. Any question about any of that? Each species has its own minimum, maximum, and optimum temperature for growing. The minimum temperature is the minimum that the species can grow. If you drop the temperature down further, the species will no longer grow. The optimum temperature is the optimum temperature where the organism can grow at its fastest rate. Microorganisms are classified into groups based on their preference for a temperature range. You can have a psychrophile, and these are organisms that grow best around 10 degrees Celsius. That's the temperature of your refrigerator. There's also psychotrophs, and these are organisms that grow best around 22 to 25 degrees. These are organisms that grow best around room temperature. There are mesophiles, and these are organisms that grow best around 37 degrees Celsius. 37 degrees Celsius is the internal human body temperature, and so all human pathogens are mesophiles. Any question about any of that? A thermophile is an organism that grows best between 60 and 65 degrees Celsius. And then a hypothermophile is an organism that grows best above 90 degrees Celsius, but generally less than 100 degrees Celsius. Hypothermophiles were not even known when I took microbiology many, many years ago. Now, you should know that the exact optimum will vary for species, and then the range especially varies for what would be the minimum temperature it will grow at and the maximum temperature it will grow at. Like Micrococcus luteus is considered a mesophile, but it grows best at 30 degrees Celsius, not 37 degrees Celsius. Why? Because Micrococcus luteus is often found, or I should say it's always found, but it's on people's skin. And so it grows best around 30 degrees Celsius. You can find it growing out in the yard or something, and actually very seldom would it ever get to be 30 degrees, but on a hot summer day, it could get up to 30 degrees Celsius. My point is Micrococcus luteus, even though its optimum growth rate will happen at 30 degrees, we still call it a mesophile. Any question about any of that? When we're talking about temperatures, you should realize that food spoilage happens when something is left at, see, I'm going to blow this up. Something is left between 60 degrees Fahrenheit to about 130 degrees Fahrenheit. That's about 15 degrees Celsius to 51 or 52 degrees Celsius. And that's because the organisms, the microbes, can grow rapidly. Some of these bacteria can even produce toxins. And so food should not be left in this temperature range for more than about 4 hours, because you can get enough bacteria growing that some of them can produce toxins. Now, food or microbes can grow at a lower temperature and a higher temperature, but they will not grow near their optimum rate. So the bacteria can grow very slowly, up to about 62 degrees, or down almost to 0 degrees. The bacteria could grow very slowly, in which case you shouldn't eat it, but you have more time. An example is the food you put in the refrigerator can remain in the refrigerator for about a week. After about a week, unless the food has another preservative in it, like pickles or ketchup have acid and salt, and ketchup also has sugar, in which case it can remain in the refrigerator much longer than a week. But most food, like, I don't know, steak you put in the refrigerator, after it's been in the refrigerator for more than a week or after a week, you should probably throw it out. And that's because the microbes could grow slowly in it. This temperature range, you can take a look at the chicken um, in the grocery store going through the rotisserie. If it looks like the chicken is looking old and starting to dry out, it's probably been there for too long, and you could get the bacteria to grow at it at this temperature range. Any question about any of that? All right. For food spoilage, if you were to put like cook up a big batch of rice and put it in a very large pan and then stick it in the refrigerator, it may not cool quickly enough because it's in such a large batch, meaning when the temperature drops from this range to this range, it can be in this 1, 2, 3, over 4 hours, and that will be enough time for the bacteria to produce a toxin, such as Bacillus cereus, which can be on rice and can produce a toxin. And so if you put it in the refrigerator in a large batch and it takes more than 4 hours to go through, I don't know, 40 degrees to about 15 degrees, 17 degrees, then it's in too large of a batch, and then the toxins can be made, so you shouldn't eat that rice. On the other hand, if you divide this rice into a smaller portion and then put several of these bowls in, like 3 bowls in, the food is in a smaller portion, and so when you put it in the refrigerator, it'll go from 40 degrees to 10 degrees or less in a shorter amount of time. Here it's under 2 hours, well under 2 hours, about under an hour and a half, actually. Um, and the bacteria will not have enough time to make the toxins. And so you should separate a large batch into smaller batches so that the food will not have enough time to make toxins. Any question about any of that? All right. So pH is another physical requirement that most bacteria have, and that is most bacteria grow best between pH of about 7 to 8. So meaning most bacteria grow best around pH 7. This can be a problem for lab cultures because if you have a lot of microbes that you're growing in the media, the microbes will tend to make acidic end products, which will then make the media so acidic that the microbe can no longer grow in it. To minimize this problem in the microbiology lab, we actually incorporate pH buffers into the culture media. And then when the acid is main, the buffers will combine with and neutralize those acids. Any question about any of that? Molds and yeast are a little different. They tend to grow best between pH 5 and 6. So they do grow best around pH 5 and 6 instead of, what is it, 6 to 8. And this could be a problem. What you could do is you could put the mold and the yeast in an even more acidic environment, and then they can't grow at least very rapidly, like be pH 4 or less. Acidophiles are organisms that grow in acidic environments, such as a volcanic environment, where the pH of the volcanic environment can be between pH 0 and pH 3. Anyone know an environment similar to that in the human body? All right, your stomach is actually an acidic environment. It doesn't go down to pH 0, but it does go down to, I believe, pH 1. And that depends on obviously what you have in your stomach. If you have food that can buffer the pH. It may not reach pH 1, but if you don't, the pH of the stomach can drop down to around pH 1, and we use that acidic environment to help digest our food. Another physical requirement that organisms have is osmotic pressure. Microbes do require a certain amount of water in order to be metabolically active. And if you have too much salt or other osmotic pressure around them, the water will leave the cell, and then that will stop the cell's metabolism if it does not kill the cell directly. Like E. coli, put into an environment with a lot of osmotic pressure, the water will leave the E. coli, and then the E. coli will quickly die, certainly within a day. I mean, I don't know the exact time, but within a day, the E. coli cells will die. A hypertonic environment includes one with higher solute concentration outside the cell than one inside the cell. And that higher solute could be more salt or more sugar. It's usually one of these two or the other. One example would be molasses, where the molasses is such high osmotic pressure, if the bacteria were to fall into the bottle of molasses, the water will leave the bacteria and either kill the bacteria or at least prevent it from growing, as well as preventing it from being metabolically active. High osmotic pressure can be used to preserve food, such as molasses. We usually don't refrigerate the molasses, but salted meats, salted fish, honey are all examples of using osmotic pressure to preserve the food. How it preserves the food is at higher salinity or osmotic pressure outside than inside, so the water will leave the cell, causing plasmolysis, where the cell membrane will shrink away from the cell wall. Depending on the cell, this could kill the cell, like E. coli, it will kill the cell. And when E. coli falls into a bottle of molasses, the water will leave the E. coli, and then that will kill the cell. Any question about any of that? All right. Halophiles are organisms that like to live in salt. Phile means lover. Halo means salt, so literally it means salt lover. There are two types of halophiles. An obligate halophile. These require high salt concentrations in order to grow. You take it out of the high salt concentration, the organism will not grow if it remains alive. There are also facultative higher halophiles. These organisms do not require the high salt concentration to grow, but they can tolerate greater than 2% sodium chloride to grow. An example of a facultative halophile is Staphylococcus aureus. It can grow nutrient agar, which of sodium, that's 0.9%. But it can also grow on an MSA plate, which has 7% sodium chloride. Much too much bacteria in order for most organisms to grow. Any question about any of that? Let's see, I was going to mention something about Staphylococcus aureus can grow on the human skin, which can reach up to 20% sodium chloride. So Staphylococcus aureus is a facultative halophile, but it can grow on like nutrient agar, which is only 0.9% sodium chloride. An obligate halophile would be like one growing in the Dead Sea, the sea between Jordan and Palestine. That sea has sodium, about 30% sodium chloride. That's the highest amount of sodium chloride you can have in a water. If you put more sodium chloride, the sodium chloride will simply precipitate out of solution. All right, any question about any of that? In a hypotonic solution, meaning low osmotic pressure, water can enter the cell by osmosis, and if the cell lacks a cell wall, the cell will have more water come in until the membrane bursts, and then that will kill the cell. However, if the cell has a cell wall, the cell membrane can only expand up until the cell wall. After that point, the cell membrane cannot expand any further. That will limit water flowing into the cell. And that's actually how most bacteria live when they're infecting a human patient, and that is they're in a hypotonic solution with with water moving into the cell. If the cell has a cell wall, it won't be a problem because the cell membrane will come up to the cell wall and then it can't move any further. That will limit the flow of water into the cell because there's no room for the water. But if the cell doesn't have a cell wall, like a red blood cell, and you put it into distilled water, water will enter the cell, causing the cell membrane to expand until it expands so much that the cell membrane pops and gets a hole in it, and then the cytoplasm can leak out of the cell, killing the cell. Any question about any of that? All right. Carbon is another chemical requirement. All organisms have to have a carbon source. Carbon is typically half of the dry weight for a cell. So carbon is about half the dry weight for all living organisms. Carbon is used in structural organic molecules. Carbon is used as an energy source, like both glucose, carbohydrates, and lipids can be used as an energy source, and they can use those molecules to get their own carbon for their carbon needs. A heterotroph is an organism that uses an organic carbon source. Humans are heterotrophs. We get our carbon needs from the carbon we eat and the carbon we drink. But an autotroph makes its own sugars, for example, its own molecules, starting from CO2 and water. An autotroph get their carbon source from CO2 either dissolved in the air or in the water. Any question about the difference between a heterotroph and an autotroph? All right, nitrogen is another chemical requirement. You don't need to know this, but about 14% of the dry weight is from nitrogen. So 14% of the dry weight of a dried cell is nitrogen. Nitrogen is found in amino acids, in proteins, DNA, RNA, ATP. Uh, Nitrogen is often obtained from the decomposition of proteins. So when an organism dies, another organism can digest those biological molecules and then get the hydrogen and carbon molecules they need for whatever they're going to be doing next. Sulfur, well, wait a minute. Most bacteria do get their nitrogen source from degrading nitrogen in a protein, but many bacteria can get nitrogen source from either ammonium ion it's ammonia with the hydrogen attached, or from nitrate as their nitrogen source. If you recall, I mentioned nitrate. Nitrate is the preferred nitrogen source for green plants, and nitrate is a significant part of your food fertilizer, your chemical food fertilizer. There are a few bacteria that can also pull nitrogen gas out of the air, pulling nitrogen gas down to fix it in an organic molecule like an amino acid. And this is called nitrogen fixation. These organisms add nitrogen to the soil or whatever they are by pulling nitrogen down from the air. And then when that organism dies, that nitrogen in the amino acids is free for another organism to utilize. Any question about any of that? All right, sulfur, another chemical source required by cells, amino acids, the B vitamins, thiamine, and biotin, as well as DNA and RNA, all require a sulfur source. Most sulfur is obtained for most cells, most sulfur is obtained by decomposition of proteins. But many bacteria can use sulfate, SO4 2 negative, and a few bacteria can even use sulfur in hydrogen sulfide gas, H2S. Phosphorus is a molecule important in cells in their DNA, their RNA, their ATP, and in the cell membrane. A phospholipid always includes one phosphorus, and that's why phosphorus is important in the cell membranes. Phosphorus chemical formula is PO4 with 3 negative charges. Phosphate is an important source of phosphorus. Any question about any of that? All right. Living elements, as far as I'm aware of, all of them require trace elements. These are inorganic elements required by microbes in small amounts. They're often required as an enzyme cofactor. Some examples of trace elements include iron, copper, and zinc. Most of you know that iron is a requirement for humans, but most of us get enough iron from the proteins we consume or the proteins we consume in liquid milk, meaning dry and liquid milk. Some examples of a trace element include iron, copper, and zinc. And most organisms get their trace elements just from the environment that they're in. Like humans get their trace elements from the food we eat, which contains the trace elements, or the liquids we drink. Very rarely, a patient coming into the hospital will be found to be deficient in trace elements, and then they'll give them copper, or whatever they need directly. But most of the time, most of us get our trace elements from the food we eat and the liquid we drink. And the trace elements are just dissolved in the medium. Any question about any of that? All right. Organisms may require organic growth factors. An organic growth factor is a compound obtained from the environment that the organism itself cannot synthesize. So with Streptococcus pyogenes, it does have an organic growth factor that must be added to the media, or else Streptococcus pyogenes will not grow. And there's a senator from Missouri who was talking about organic growth factors. Well, let's go back to Streptococcus pyogenes. It requires the V factor and the, is it F factor? Which is, one of them's NAD and the other is, it might be biotin, a B vitamin. But Streptococcus pyogenes can get those from blood if we add blood to the media. And Streptococcus pyogenes can lyse the red blood cell. And then that would release the organic growth factor that is required by the cell. Some examples of organic growth factor include vitamins, certain amino acids, we call them essential amino acids, and certain fatty acids, we call them essential fatty acids. These are molecules that we cannot build ourselves. Why they're called vitamins actually is because humans can't make them, but we require the vitamins. Certain purines and pyrimidines may also be an organic growth factor. If the organism requires a purine, then you must include it in the media or the organism will not grow. Any question about any of that? All right. Molecular oxygen is, in a sense, a toxic substance. There are 4 toxic forms of oxygen, which never are O2 as found in gas. All of them are usually intermediates of some chemical reaction, such as an intermediate in aerobic respiration. There are 4 toxic forms of oxygen. One is singlet oxygen, where we have oxygen gas, and then the electron, or 2 of them, depending on what's happening, absorbs some energy, and then the oxygen becomes singlet oxygen, which can damage the cell. Singlet oxygen is one where the oxygen atoms are elevated to a high energy state. Even white blood cells, which have some non-specific defenses for cancer, can use singlet oxygen to then secrete that against a pathogen or something that's in the body, and the white blood cell goes up to it. It can release the singlet oxygen onto the pathogen with the intention of eventually killing it. Any question about any of that? Oops, what did I do wrong? Another toxic form of oxygen is superoxide free radicals. This is an oxygen molecule with an extra electron, and that's what we're showing with that minus sign there, as well as that cube. That means an extra electron, and it has a -1 charge. So that would be a superoxide free radical, a oxygen molecule which has an extra electron, giving the molecule a -1 charge. That symbol, which is sort of like a cube, is referring to an unpaired electron, which unpaired electrons are only found in free radicals because the cell wants to keep the electrons paired. And that's just actually normal chemistry, that the electrons want to be paired. And if ever they're not, then you have a free radical because that molecule can come bump up into something and then steal an electron from that other molecule. Superoxide free radicals are formed during normal aerobic respiration. Superoxide free radicals can steal an electron from a intermediate of the electron transfer chain. And then the molecule that donates its electron to the superoxide free radical can then bump into another molecule and then steal an electron from that molecule. Pretty soon we have this going on all over the cell, and there's all sorts of radicals, or what used to be a free radical, on the floor, and someone will finally realize that you don't want to keep accumulating more and more of these superoxide free radicals. So the cell will neutralize superoxide free radical with the enzyme superoxide dismutase, or SOD. So we have two superoxide free radicals, combine it with two hydrogen ions in the presence of SOD, the enzyme, and then you convert this molecule into hydrogen peroxide and water. And it's true, hydrogen peroxide is also toxic to the cell, but the cell probably has a means of removing hydrogen peroxide from the cell. And that's why the cell can grow in the air, because it has a way of dealing with hydrogen peroxide. So hydrogen peroxide is a peroxide anion. Specifically, a peroxide anion is O2 with two extra electrons, giving it a 2 negative charge. This molecule can combine with two hydrogen plus ions to make hydrogen peroxide. Hydrogen peroxide is toxic to cells, but most cells have a means of neutralizing hydrogen peroxide. If a cell has catalase, the catalase can take the two molecules of hydrogen peroxide to make two molecules of water and one molecule of oxygen gas. And so catalase can neutralize the hydrogen peroxide. Most cells that live in the air, meaning aerobes, have the enzyme catalase. It's not true that all cells do. There's R2 that we mentioned in our last lab that do not, but most cells that live in air do have the enzyme catalase. If they do not have the enzyme catalase, but they're living in the air, then they must have the enzyme peroxidase. Peroxidase can once again take hydrogen peroxide, combine it with two hydrogen ions in the presence of peroxidase to make two molecules of water. So you're converting the substance from a toxic substance to actually a beneficial substance. Any question about any of that? All right, the fourth toxic form of oxygen of the four is a hydroxyl radical. Let me blow this up. So here we have the hydroxyl group, and it has this unpaired electron there. Note that that does not have a charge to the hydroxyl radical. But this free radical can go up and bump into any molecule and then steal an electron from that molecule. If it were to bump into DNA, it could mutate the DNA just by stealing the electron from the DNA. So do note the hydroxyl radical is different from a hydroxide ion, that's OH with a -1 charge. And it's also different from the hydroxyl group, like an alcohol, in a carbon molecule, meaning a carbonic molecule. Hydroxyl radicals are always made by aerobic respiration in trace amounts. They're also made by ionizing radiation. And when hydrogen peroxide reacts with certain metal ions, you can make hydroxyl radicals. There are antioxidants in the body, at least the human body, to react with and neutralize the hydroxyl radical. So that's how we as humans deal with the hydroxyl radical, we use an antioxidant such as vitamin C or vitamin E, which combine with the hydroxyl radical and then tie it up, and it's no longer a radical. That does use up the vitamin C and the vitamin E. I mean, you can't use them as a vitamin, but they have been used for a good purpose, and that's to neutralize and tie up the hydroxyl radicals. All right, any question about any of this so far? Microbes that use oxygen, we call them aerobes, and they produce more ATP from nutrients than other organisms that do not use oxygen. Microbes are classified on the basis of the effect oxygen has on their growth. So the easiest one to explain is an aerobe. These oxygens, when you grow them in a thioglycolate tube, thioglycolate media itself pulls out oxygen from the media. So once you first take your tube of the thioglycolate media out of the autoclave, there will be no oxygen in the thioglycolate media. However, the air is about 20% oxygen, and it will come in to the thioglycolate media, and at the very top of the tube, the oxygen amounts in the top of the tube will be 20% or very close to it. Like 20.01% or something like that, 19.991%. So at the top of the tube, we'll have full oxygen. At the bottom of the tube, we'll have no oxygen. And as you go down the tube, you'll have less and less oxygen, meaning an oxygen gradient will be established in the tube. When you inoculate the tube, thioglycolate tube, with a aerobe, then the bacteria will only grow in the top of the tube where there is oxygen. And as you go down the tube, you'll see less and less growth. So like we said, it should have one right there. You'll see less and less growth. And the reason for that is because there's less and less oxygen the further you go down the tube. And eventually, in the end of the bottom of the tube, there will be no growth because there is no oxygen. Any question about an aerobe and how it grows in the thioglycolate media? If next, let's talk to C, the obligate anaerobe. These are organisms that only grow in the absence of oxygen. So you'll see most of the growth in the bottom of the tube where there is no oxygen. And as you go up in tube, you'll see less and less microbes because there will be more and more oxygen, which that would be toxic to the cell. The next easiest to explain is probably the facultative anaerobe. This is an anaerobe, so it can grow in the bottom of the tube, but it can grow in the top of the tube. And a facultative anaerobe is one which can engage in aerobic respiration if oxygen is present, but if oxygen is not present, then the microbe will rely on an anaerobic source for its needs, for its nutrient and energy needs. So because it grows best in oxygen, you'll see the most growth at the very top of the tube. And as you go down the tube, you'll see less growth than at the top. But when you go all the way down, you'll always have growth, about the same amount on the bottom. And that's because these organisms can grow in the absence of oxygen. That's why they're growing at the bottom of the tube. But they can grow in the presence of oxygen. In fact, they prefer it. And that's where they'll have their most growth because that's where the optimal growth is, at full oxygen. All right, then we have an aerotolerant anaerobe. This is an anaerobe. So it does grow in the bottom of the tube where there is no oxygen. But this aerotolerant anaerobe can tolerate oxygen because it has all the chemicals and all the enzymes it needs to remove the toxic forms of oxygen. And so an anaerobic aerobe, or aerotolerant anaerobe, I guess is what I'm trying to say, can grow everywhere on the tube. And it will grow everywhere the same because it doesn't need or use oxygen, but it does have the enzymes it needs to remove the toxic forms so they don't kill the bacteria. And then we have another one, a microaerophile. These are aerobes, so they do require oxygen in order to grow. But these microaerophiles do not like growing in full oxygen. They like growing someplace between 2 and 10% oxygen. And so you'll see the best growth in the middle of the tube, where the oxygen level is reduced, but not down to zero. And there's more than 20%. There's less than 20% because that's there. And as you go up the tube, you'll actually see less growth than in the middle of the tube. And as you go down the tube, you'll also see less growth. And the reason for the less growth is because there's less oxygen than in the middle of the tube. Any questions about the effect oxygen has on microbial growth? All right, if there's no questions, I'm going to stop here. I will be showing up in the lab for the first 15 minutes. We have no lab. I'm only going to show up to answer questions. And after the first 15 minutes, or the last question is answered, I will log out. So if you have a question, show up in the lab. If you don't have any questions, you do not need to show up in the lab. Oh, come on. All right, goodbye.